Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science podcast. I'm Ayesha, Senior Life Science Journalist at Xtalks.com, and this week I'm happy to introduce a very special guest on the show. We have with us today Jerry McLaughlin, who is the CEO and board member of Life Biosciences, a company advancing innovative cellular rejuvenation platforms to reverse diseases of aging. Jerry has over 30 years of experience in the biopharmaceutical industry and has been involved in the discovery, clinical development, and global commercialization of more than a dozen FDA-approved drugs with multiple successful exits. Jerry began his career at Merck and was extensively involved in multiple blockbuster product launches. Life Biosciences is developing a gene therapy for two types of optic neuropathies with significant unmet needs. To learn more, let's turn to Jeffrey now. Thank you so much for being on the show today.
Thanks, Ayesha. It's a pleasure to be here and looking forward to discussing Life Biosciences today.
And we are looking forward to hearing about all of the exciting work happening at Life. So to begin, I'd like to ask, what are the mission and goals of Life Biosciences?
Yeah, our biggest mission and what we focus on every day is making a difference across the world for patients and humans who are at risk and experiencing age-related disease. As we all know, there's a huge global population with today with depopulation, and it's more imperative than ever that we live healthier, longer lives. And the way we're doing that is by developing therapies that target the underlying aging biology for these age-related diseases, and that's a relatively new concept in the pharmaceutical industry. We focus on restoring health span and extending health span. And what that is, the difference between that and lifespan is it's the number of healthy years in one's life. And so the idea is live healthier longer and you live more productive and a happier life as well. We know that historically aging was just thought to be random wear and tear. And it's never more present than in our political scene in the United States, talking about the different ages and the health of individual candidates. But we know age is a number, and it's really about how healthy you are at that stage in your life. And in addition, the pandemic really brought an awareness to the general public about aging, and it's a risk factor for disease, and in that case, COVID. That coupled with advances in science, because I get the question often, why haven't we able to do this before? Well, technology, better chemistry, has allowed us to really identify these underlying drivers of aging, and one being epigenetic changes, which we'll talk about, others proteostasis, and there are others, there are about 12 in total. And we have a number of companies, including Life Biosciences, who are attacking these core underlying drivers of aging to see if we can, in fact, reverse and prevent disease. We're moving away from just treating symptoms of disease. And in fact, when I started at Merck 34 years ago, we were bringing novel medications to the market to treat a variety of chronic diseases of aged persons. I think the big difference today, as I look back, is we were targeting receptors and symptoms. And so now we have an ability to target that underlying cause, which gives us more opportunity to not only treat, but to reverse or prevent. So that's the status of it. And so when we look more closely here at the epigenetic reprogramming, scientists have identified that you can have modifications to the epigenome, such as DNA hypermethylation. That what they do is they modify the expression of genes by increasing or decreasing their expression without altering the genes themselves, which is quite fascinating and a relatively new concept. And the epigenome drifts with age, leading to just dysregulated gene expression and making our bodies more susceptible to disease. The good news is we now know we can restore. Unlike once genes are altered, the epigenome can be restored to a more youthful, and resilient, and productive state. And that's the basis of our company, as you mentioned earlier, focusing on cellular rejuvenation. We have the platform you had mentioned, partial epigenetic reprogramming. We also have a program called Chaperone-Mediated Autophagy, which seeks to restore proteostasis, and we can talk about that a little later as well. Our lead programs are into optic neuropathies, which I think are a good blend. One is an acute ischemic event, also known as a stroke in the back of the eye, called non-arteritic ischemic optic neuropathy. And the other is a very, very common, well-known chronic optic neuropathy called primary open-angle glaucoma, which affects more than 3 million Americans currently.
Indeed, and significant unmet needs in both of those indications. And so can you tell us about how you're really targeting the underlying causes of these diseases, of these particular neuropathies? As you mentioned, again, that it's not about treating the symptoms anymore, and that really the goal is to target the underlying biology and the pathophysiology of these diseases. And so what is, number one, the underlying, in essence, like the underlying causes of these diseases? And then how does Life Biosciences come in with what it's doing to target them?
Yeah, so the underlying biology really focuses on, in these optic neuropathies, is damage and death to the retinal ganglion cells in the back of the eye. This is a common site of degeneration in older adults, and there's an area where there's also been precedence for gene therapy. So for us, it made a lot of sense as a place to start. It's a well-contained area of the body from a safety standpoint, but also an area that has been studied before, and a clear driver of this morbidity in the retinal ganglion cell dysfunction. In the case of NAION, you have a decreased blood supply to the optic nerve, which can result in the effect and the injury to the retinal ganglion cells. And with glaucoma, it's just the long-term intraocular pressure that is leading to slow, insidious degeneration of these retinal ganglion cells. And so it really begs the question: what if we could preserve or restore retinal ganglion cells? And that's really the basis for what we're doing. And when you look at the numbers, NAION, as I mentioned, is small. It's really an orphan indication in the U.S. Roughly anywhere from 2.5 to 10 out of every 100,000 Americans will experience NAION in a given year. So it relates to anywhere between a few thousand to 10 to 15,000 patients per year. It's the most common acute optic neuropathy in patients over the age of 50 years of age. It's a little scary in that it's often painless in terms of its onset, and often happens in the morning upon wakening, where an individual will wake up and they can't see out of one eye completely, or at least one quadrant of one eye, and it can be quite scary. The other issue with this optic neuropathy NAION is that once you have it occur in one eye, you have a 20 to 30% chance over the next 5 to 10 years to experience in the other eye, and that can have a significant impact on quality of life, losing not only vision in one eye but substantial vision in both eyes. In fact, there are many studies that show losing your sight is second only to Alzheimer's and concerns among aged persons. So it's really important here that we seek ways to preserve eyesight. There are no treatments currently available at all. And we believe that that with our targeted therapy for rejuvenating aged and injured cells provides such a novel approach, and really a first approach that tries to get at the root cause of the damage. And so we think this holds promise for this population that today is basically told improve your lifestyle to help prevent it. And that's the really only treatment today. What's interesting with NAION, just in the last week, you may have read in the news, the ever popular GLP-1s. A large retrospective analysis came out that showed there's a link, a significantly increased risk of patients experiencing NAION who are also on GLP-1s. And that is an important finding, in that the number of individuals on GLP-1s today, it's estimated 6% of the adult population is currently taking GLP-1s in the U.S., which is roughly 10 to 15 million. And that number will rise anywhere to 30 to 50, even 70 million based upon forecasts over the next 5 years in the U.S. So that will actually be the single largest driver of NAION, given that data. And we think it's an important concept, right? Where patients, while the data did not show a direct causal relationship with GLP-1s, there's no doubt there's underlying biology that would make individuals over the age of 50 who have a predisposition and are on a GLP-1 more susceptible. And that's what the data suggests. So it's an important consideration for anyone on a GLP-1. And so we think that just even heightened our urgency and the importance of our work going forward, because we're not here to say GLP-1s are bad. They're amazing, amazing, game-changing medications and showing all kinds of benefits. But it really highlights the need to look at things that are associated with this large class of medications, which include other things, includes muscle wasting, right? And there are efforts now to overcome that as well. So we're real excited about the ability to help and move this forward as fast as possible. And then in our other indication, primary open-angle glaucoma, as I mentioned, it's very, very common. We all know people with glaucoma, 3 million patients in the U.S. It is the leading cause of blindness for people over the age of 50. And unfortunately, while there are many treatments for glaucoma, both surgical and therapeutic, the goal or the outcome has not changed in the last 50 years. Basically, everything is about either reducing intraocular pressure or relieving that pressure by creating avenues for aqueous to drain, and so that's the only the standard of treatment today. And so our belief is if we can, for many patients with glaucoma, particularly as it advances when you have more retinal ganglion cell death, if we could preserve and restore retinal ganglion cells to a more youthful state, we have an opportunity here to change the game, right? And either alone or in combination with the current therapies. And I can tell you, when I started at Merck in 1990, we were selling a beta blocker for glaucoma. There's still frontline therapies today, and so we really haven't made much progress on that front. But I think with the science today, it's really given us an opportunity to make a really big difference in the world.
Mm-hmm. Indeed. And so that's really exciting to learn about how relevant these pathologies, these diseases are, and in the context of, as you mentioned now, with GLP-1 drugs and an aging population and all of that. So it's very relevant to be pursuing drug development in areas with so much unmet need. And like you said, with drugs that have been used for decades and no new treatment. So that's very exciting. And so I'm looking forward to learning more about your lead compound, ER100. And so could you tell us a bit more about that and how it's being developed? And of course, it's based on the partial epigenetic reprogramming technology at Life Biosciences. So love to hear more about the compound and how it works, and also at what stage of clinical development it's at, or preclinical development, and
Sure.
all of that.
Sure. So I think to get to where we are today, I think a little history lesson here for everyone is that looking back to the onset of understanding of transcription factors and their ability to reprogram aged cells. Dr. Yamanaka from Japan first published data in 2006, and it really turned the whole field on its head. And what he was able to show, in fact, he won the Nobel Prize in 2012. He discovered that 4 transcription factors, and I'll just say them, OCT4, SOX2, KLF4, and c-Myc, could reprogram mature differentiated cells back into stem cells, back to their pluripotent state. So fully reprogram, which was absolutely scientifically amazing. Scientifically amazing. However, translating that discovery into a therapeutic, there are several limitations. And those limitations are twofold. One, c-Myc is known to drive and cause teratomas, number one. And number two, when you fully reprogram a cell back to a pluripotent state, it loses its cellular identity. And in a very practical term, it may forget whether it's a kidney cell or a retinal ganglion cell. And so that's really problematic, and that's not what you want. But it was Yamanaka who really set up the entire field to determine, now, how do we do this in a controlled and precise fashion? How can we restore these to a youthful state, but not to a pluripotent state? And that's where David Sinclair came in from Harvard, our founder of our company and a tremendous scientist in the field of epigenetics. And what he discovered was that by only turning on 3 of the 4 factors, OCT4, SOX2, and KLL4, that is the sweet spot for developing therapeutics where you can partially program, and it's estimated bringing them back about 80% of the way, right? And restore cells to this more youthful state. And the great news is they maintain cell identity. You don't see formation of teratomas even over the long term. And so that gave us the confidence then to move this into animal studies. And what was demonstrated in multiple mouse models of aging and optic nerve crush and glaucoma is that in an aged model, we see natural age decline or a vision decline in aged in mice. And with the introduction of OSK, these 3 Yamanaka factors that we now call ER100, Dr. Sinclair was able to see improved vision and reversing this DNA hypermethylation I'd mentioned earlier, right? Eliminate some of these marks. So you're restoring this epigenome back to what it was when it was younger. And so that was done in an aging model. Then in an optic nerve crush model, never before in humans and animals, within days after when we're born, we cannot create new nerves. What David was able to demonstrate in an optic nerve crush model in the area post nerve crush was able to promote axon regeneration, reverse DNA hypermethylation, and actually increase retinal ganglion cell counts. So really phenomenal groundbreaking work. And then just to take it to a more practical standpoint, in a glaucoma model in mice, was able to improve pattern ERG, which is a measure for visual function, and improve vision overall by an optomotor response tool. And so you had this perfect storm here of natural aging, vision loss, restored to a younger state, regenerate nerves and retinal ganglion cells, and then in a glaucoma model, show visual restoration, which is all fantastic, right? And so the next step is big question when you're talking about eye disease. The human eye and the mouse eye have not much in common, and structurally, at least that's important for developing therapeutics in our situation. The big challenge and the big step forward for us was to conduct studies in non-human primates. And we could talk about that a little more later, but that was a big step forward because non-human primates, and particularly these Afro-Green monkeys that we used, they have an inner limiting membrane, which is a barrier to delivering therapeutics. And so we were able to move then on to these non-human primates and demonstrate restoration of visual function, and also, we believe, restore or preserve retinal ganglion cell counts, healthy retinal ganglion cell counts. So that's our program we call ER100 that induces this partial epigenetic reprogramming to modify the epigenome, once again, only using 3 of the 4 Yamanaka factors. And it's different, and why we call it is a gene therapy, it's different from what most people think of as common gene replacement or gene editing approaches, in that we're not, it's really important, we're not altering GNAD.
Right. We're not editing the genome or anything like that.
No, we're not. Yeah. We're not. We're bringing the software package back and debugging it, putting it back to its, making our iPhone work faster.
Right.
So.
Yeah, that's wonderful.
And I think you had asked about the stage of development of where we are.
Yeah, like you did start talking about some of the promising data and in the non-human primates, which was
it was really the big step forward, right? Because it really is that bridge, bridge between the early animal work and in the eye, human clinical trials, and so we talk about NAION, this non-arteritic anterior ischemic optic neuropathy. There's actually an exquisite model to use, which is very important in drug development. You want a model that replicates and the human condition as much as possible. So it's a laser model, laser-induced NAION, and so what we were able to demonstrate, what you see in human NAION is when you see a diminished pattern ERG. And you see your peak to trough ratio in your pattern ERG, it shrinks, and that is a great measure for visual function. In the model, we were able to induce in these non-human primates, induce NAION and measure their pattern ERG changes, and it was highly comparable to the human condition. And so because of that, we feel very confident that the results would be a good predictor in the human condition. And so what we did, we took a finite number of primates, 6 and 4, I believe, in each group, both active and placebo. And we induced NAION, and then we took pattern ERG at baseline, and then we took it at several time points over a 42-day period. And we actually saw significant improvement in pattern ERG, even at those small N, in the group treated with OSK versus those on control. Which gave us great excitement to move forward. It's we believe the first of its kind in a epigenetic reprogramming in non-human primates, and gave us that confidence then to engage in a manufacturing campaign and to move on to meet with the FDA and move on to IND enabling work. So with that validation in hand, we are rapidly advancing toward the clinic, with plans to be in the clinic as early as the second half of 2025. In fact, in our package, in our IND enabling package, we just dosed a key new study just this past week in one of the IND enabling packages. So we're excited about that, and we're really excited about where we can take this going forward. And once again, getting back to this fact that this has never been shown before, right? That when you're altering the epigenome, that through cellular rejuvenation, you can restore visual function. And I think it really puts us in an exciting place going forward.
Indeed, and it's always wonderful and it's very exciting to hear about the applications and the translation finally to human diseases. Going through school, grad school, studying epigenetics, studying all of these things, but now to see them being developed into actual therapeutics that can have an impact on human health, it's just amazing. I think it really comes full circle, and it's so exciting to really hear about the work that you're doing at Life Biosciences. That epigenetics is just not like a chapter in a biology textbook anymore. So to really
it's an industry now.
It's an industry now.
There are no fewer than a dozen companies. We're flattered. We think we were one of the first.
Yeah, indeed.
And we believe we're the furthest along.
Yeah.
But there are a number of new companies, and we applaud them, and we hope they're successful because we think this is an exquisite way to target disease.
Absolutely. And so for ER100, are there other indications that you're planning to look at it in? I know that you have the 2 eye indications, but could it be explored beyond the eye or other eye diseases?
So yes, and we don't disclose those, but I can definitely tell you, we think there's more opportunity within the eye, even targeting retinal ganglion cells, but looking at RPE cells. In the eye, we think there's opportunity because what we're doing for, and once again, I think it's important, I forgot to mention this with the delivery. Our ER100 is delivered through intravitreal injection. So it goes into the vitreous and then migrates across the inner limiting membrane to the retinal ganglion cells. That's the target here. And we've been able to show expression there, obviously, with it working. But there are other ways we can go. We can go subretinal and target other cells. So we're looking at not only new and additional indications in the eye, looking for different deliveries. We're looking beyond AAV. We're going to use the best vehicle to get OSK to the target tissues and organs and cells. And so we see that there in the eye, but then beyond that, there's been a number of work done in neurodegenerative diseases, such as Alzheimer's, frontotemporal dementia, other ophthalmic disorders, as I mentioned. And we think a laundry list of target tissues and organs where you have aged and injured cells. If we can restore them to a more youthful state, we can reverse disease and prevent additional disease progression. So we think we're just at the beginning. The unmet need is compelling in these optic neuropathies, but it's really setting the stage for the future as well.
Indeed. And so I know that you did mention how you've seen this space evolve over the past 3 decades. And so what are some of the things that really stand out? You did touch upon them, and leveraging your tremendous and extensive experience in biopharma. How do you see the industry having developed or evolved, and where do you see it evolving to in the coming years the next, let's say,
I think that's a
3 to 5 years?
a critical question.
Yeah.
And we're living in the evolution of this category for a while, and it's a category that it holds a lot of consumer and general interest around longevity and living forever, and that's the easy sort of answer, right? The easy marketing thing, and so what we've done, and the team we have, we focus expressly on rigor, quality of data, FDA regulations. Diseases with indications that have significant unmet medical need, and so I think you've seen I think we're not the only ones, but there's been a marked aim to evolve this field into more traditional biotech science and drug development, discovery and development. I think in some of the early days there was a hope that we could develop the magic pill or the magic injection that would get an aging claim for folks to live longer. I think there's a path to that, but there's a reality of regulatory reimbursement, drug development limitations, cost that really put us on a path where we can help a lot of patients live healthier, longer lives, starting out with targeting individual indications. And I think that's the stepping stone approach that will work. It's the tried and true method. Bring value to patients, then demonstrate with additional data and broaden your indications and broaden the evidence for the field. And ultimately, there may be chances for these more longer-term indications, right, around mortality and living longer. But I think really focusing on rigor. And we built our team here. Our chief scientific officer brings 25 years of biotech and pharma experience in drug discovery and development. We bring professional CFO who's 20 years in biotech as chief financial officer. On the legal front, the same thing. And we're building a team of experienced biotech scientists from molecular biology to gene therapy. And to us, that's been the big evolution in this sector. And I think building credibility. And the second thing is, the proof is in the data, right? Always, right? You're interested to know when's that data, when's the human data coming? We're seeing a lot more companies are making their way through drug discovery and IND enabling and on the precipice of entering the clinic. And I think that's going to be another big watershed event for this class is delivering on human clinical data.
Mm-hmm. Well, Jerry, thank you so much, number one, for the amazing work that you are leading at Life Biosciences. It's tremendous. Again, I'm just awestruck in terms of like seeing these concepts being brought to life, essentially, and that can have a real impact on human diseases. And then in the end, longevity, like you said, like the primary goal is to really look at the biology and treat disease. And if longevity and living, not forever, but let's say, finding some kind of fountain of youth along the way or whatnot is in there. That's interesting as well. But I really, really appreciate your insights and working in such an exciting field and really, really being a pioneer in it. So thank you so much for your time today.
Oh, thank you for your interest, and it was a pleasure.
Likewise. That's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we will see you next time. Thank you so much. Bye for now.
To join the Xtalks community, visit Xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.